So today I'd like to give you a short explanation on where to find tardigrades. Tardigrades are also known as water bears or moss piglets, and sometimes they cannot be quite easy to find. So, hello and welcome, Microbehunter here, Oliver here, and I did receive again a few questions from some of my viewers of my YouTube channel, and some people seem to have a little bit problems finding tardigrades. These are quite well-known and cute-looking micro animals. They're pretty, interesting to look at. They have 8 legs and are microscopists' favorite animals. However, they might be a little bit difficult to find sometimes. They can be found in moss, hence the name moss piglet. However, some people have had a problem here, and I'd like to read out the question first, and then I'll give you my take on the whole issue. As a matter of fact, there are 2 questions here. I have a question. Will I be able to find a tardigrade from the moss of my garden? Because I checked it and there was no tardigrade, and not just that, there wasn't anything living. And then there's a second question here from a different viewer. I have been looking for tardigrades off and on for more than 6 months for an art project. I have looked at moss on bricks, in forests, on fallen trees, and on lichen, on tree branches, and on the roof. I put the moss in petri dishes wet and I waited. I find loads of nematodes, rotifers, and other tiny critters, but no tardigrades. I'm beginning to think of them as a microscopic Bigfoot. How do these supposedly very common animals keep evading me? Any ideas? Well, of course, thank you for the questions here. Common question. Tardigrades are pretty popular specimens, microscopic specimens, but sometimes a little bit difficult to find. Now, I cannot guarantee you that my tips will actually make you find tardigrades, but maybe they can increase the probability here. So first of all, it's important to understand that when the moss becomes dry, then the tardigrades also start to desiccate, and they are able to withstand dryness quite well. But what I suggest is that you take the moss sample and you really make it quite wet. Not just a little bit moist, but quite wet. Of course, you do not want to completely soak the moss in water, because it does need to be in contact with air so that photosynthesis can take place. Carbon dioxide in the air is actually absorbed by the moss as well, so you don't want to drown the moss either. But if you keep it quite moist, then the tardigrades should come back to life again, and maybe they're even able to start to reproduce. So I suggest the first thing that you try is that you take a moss sample and then make it wet, keep it in a petri dish, and then try to rinse out whatever you find from the moss. So my suggestion is that you then turn the moss around, dip it in water, and agitate it a little bit and move it a little bit so that any tardigrades which might be in the moss are then washed off into the water of the petri dish, and then you can try to put that under the microscope. Now, I would say that if this doesn't work, then there were no tardigrades present in the first place. And this could be due to probably high temperature, because tardigrades, they are able to withstand cold temperatures, and they're also able to withstand dryness, but they're not very good in withstanding high temperature. And for this reason, if the moss was now in direct sunlight, and if, let's say, it was on the roof of a house or on the road somewhere, and it became dry, then the heat might be actually enough that it killed off any tardigrades that were on there. And then even if you make it moist again, then they're not going to come back to life. And as a matter of fact, I did find a scientific publication, and I'm just going to read off now from the abstract of the scientific publication, because some of the scientists, they did some research concerning the temperature tolerance of tardigrades. I'm quoting now. We estimate that the 50% mortality temperature for non-acclimatized active tardigrades is 37.1 degrees centigrade. This basically means that at body temperature, at human body temperature, 50% of the tardigrades will die. I mean, this is really not very temperature resistant here. And it goes on with a small but significant increase to 37.6 degrees following acclimatization. So this basically means if the tardigrades become used to the higher temperature, then more of them are able to survive. And desiccated specimens, these are the ones that have lost the water, they tolerate a much higher temperatures with an estimated 50% mortality temperature of 82.7 degrees centigrade following 1 hour exposures. So after 1 hour, the dry ones are able to survive. If half of them are able to survive 82.7 degrees, but with a significant decrease to 63.1 degrees centigrade for following 24-hour exposures. And 63 degrees, I mean, that is something that I would say during sunlight, the road or a rooftop can easily achieve. After all, those surfaces become quite hot. And this can actually, I think, really dramatically reduce the number of tardigrades that you find there. And last comment here, or last quote here, our results show that metabolically active tardigrades are vulnerable to high temperatures, yet acclimatization could provide a tolerance increase. Yep, so it's kind of a little bit summary here. It was an abstract. So with this knowledge, I suggest that you take the moss samples from places that do not reach very high temperatures. So moss samples that were maybe in a cool environment the whole time, maybe next to a water, maybe next to running water or in a forest. And what I've also found out is that in many cases, the tardigrades with their little claws, they like to cling to the moss. For this reason, they cannot be rinsed out quite easily. So this means that sometimes a moss sample might contain a lot of tardigrades, but maybe not, they might not be washed off so easily. So this is something that you have to consider. And last but not least, we have to take into consideration that you might need to be simply patient. I mean, maybe there are plenty of tardigrades in the water sample that you want to observe, but maybe they simply are not right now visible on your microscope slide because you only took a small amount of the water. Yeah? So this is also something that you might take into consideration. And my suggestion is that if you rinse out any tardigrades or any organisms from the moss sample under a Petri dish, maybe you want to first look at the Petri dish using a low magnification, and don't forget one thing, that if there are a lot of other particles in the specimen sample, the tardigrades, they like to cling to it, so it's kind of easily it can be easily overlooked. And going back to one of the questions here, the first question, because I checked it, I'm quoting now again, and there were no tardigrades, and not only that, there wasn't anything living. And this is already an indicator that if you don't find anything living, then this is actually quite surprising. Then, not surprising, but this actually might mean that either you did not give the moss enough time to regenerate using moisture, or indeed, maybe due to high heat exposure, many of the microorganisms died off. Yeah. But when you add water, then normally many of them might come back to life again. And so these are just some basic recommendations here. Don't give up, be patient. And I can confirm that depending on where you take the moss from, there can be quite a lot of tardigrades, or relatively few. So a lot depends on the place where you take the moss from. I think I'm going to leave it at that. I hope that you like these type of short answer type podcast episodes. Happy microbe hunting as always, and see you around next time.